7 people online, so I have no idea what turnout for these usually is. I'll just go ahead and get started. If anybody comes in, they'll come in. If you already know MEG and you're already doing MEG, I want to apologize because you really didn't need to come to this talk. But that's okay. I don't think you're going to see anything that you haven't seen before. But welcome, everyone. I'm happy that everyone's here to learn a little bit more about magnetoencephalography or MEG. I'm going to give you a little background on what MEG is and what it measures, and some examples of things that MEG can do, and where MEG is hopefully going in the future, as well as what resources we have here at the NIH. Okay, so first of all, what is MEG? So in brief, MEG is the measurement of magnetic fields outside the brain. And where do these magnetic fields come from? So the largest signals, although by no means the only signals that we measure are from the cortex, from cortical pyramidal cells. And you can see from these images by Ramoni Cajal, the rainbow technique and electron microscopy reconstructions, the cerebral cortex consists of many cortical pyramidal neurons that are oriented in parallel. Oh, and I also want to say that if anybody wants to stop, interrupt me, please go ahead. This is very informal. There are 2 types of relevant signals that occur in these neurons. There's action potentials and there's postsynaptic potentials. Now, action potentials propagate down the axon and they're quadrupolar in nature. They're very short and they're less likely to be synchronized. So those are unlikely to constitute a large portion of the MEG signal, but they may contribute to very fast high gamma signals. Postsynaptic potentials, on the other hand, are dipolar in nature. They tend to be synchronized across large populations of neurons, and they last on the order of tens of milliseconds. So this is primarily what we're measuring with MEG. So in a single cortical pyramidal neuron, inputs set up a current sink and a current source, which creates a dipole. This dipole will generate a magnetic field, and when the field from many neurons is summed, it will generate a magnetic field that may be measurable outside the head. So this is just a cartoon of the cortical surface showing the dipoles oriented perpendicular to the surface of the cortex. So this is the cortical ribbon right here. And I used this to illustrate an important point about the signals detected by MEG. So for a dipole oriented radially, you look at the pattern of the magnetic field, measurable field from that dipole will largely be contained within the brain. In contrast, for a tangentially oriented source, the magnetic field outside the brain will be larger. So EEG, which is represented by these little electrodes here on the scalp surface, will be sensitive to both of these sources. However, EEG is hampered by the fact that the scalp conducts electricity. So that hampers the spatial specificity of the signals. So in contrast, the magnetic permeability of the tissue, biological tissues, is similar to that in empty space, so signals won't be distorted. Now, even though I know here that MEG is more sensitive to tangential sources, it's really important to note that there are very few truly radial sources. And it's really only a couple of millimeters along the very apex of the gyrus that's not measurable by MEG. And if you look at the entire surface of the cortex, only 5% of the cortex is within 5 degrees of radial in orientation. So depth is really more of an issue than orientation of sources, and detection is really a signal to noise problem. So how does MEG measure these fields? So they typically use a magnetometer or gradiometer pickup coils. So a magnetometer will be a single loop, a gradiometer will be 2 or more loops, and in a gradiometer, what's measured is this difference between the field and those loops. And these coils are coupled to a device called SQUID, or superconducting quantum interference device. So the primary drawback of these sensors is that they rely on the quantum mechanical properties of superconducting loops, and they require immersion in a liquid helium bath. So this is the cross-section of a MEG instrument. You can see the helmet down here with the sensors. And most of what you see when you see this giant MEG instrument is really a thermos to hold all the liquid helium to keep everything cold. And as you can see, because there's this liquid helium bath, there has to be a lot of thermal isolation here. So the sensors actually end up being pretty far away from the surface of the head, which is something that I'll talk about a little bit later when we talk about some of the future of MEG. So now I want to talk about what things are possible to do with MEG, with a particular attention to what MEG can do that other techniques can't, as well as some common misconceptions about MEG. I'm going to use some examples from some studies that the MEG Corps has done over the years, although I'm not going to get into any study in too much detail. So we've all seen these graphs of the different neuroimaging techniques plotted in terms of their spatial resolution versus their temporal resolution. So most hemodynamic-based methods have low temporal resolution due to convolution with the hemodynamic response function. Now, I know there are newer functional imaging techniques with MRI that may have faster temporal resolution, but at present, those aren't widely available and aren't fully understood. So right now, I'm just going to leave it that fMRI has relatively low temporal resolution. But fMRI in particular has very high spatial resolution, and the achievable spatial resolution is constantly increasing as new technology is developed. Now, EEG and MEG, which are frequently incorrectly grouped together on these graphs, both have excellent temporal resolution. Now, EEG has relatively poor spatial resolution, as I talked about, in large part because of that conduction of the field of the electric potentials by the scalp. But the resolution of MEG is on par with that of PET, and in some cases a little bit higher. And although MEG does have better spatial resolution than EEG, that requires solving an inverse problem, which is ill-posed. So we have, in our instrument at the NIH, we have 275 sensors, but we want to solve for every location in the brain. So this problem is vastly underdetermined. So there are infinitely many solutions, is what I mean by this. So we have this data, which is the shadow, and like this shadow of a rabbit, you don't know if it's caused by an actual rabbit or if it's caused by someone's hand. Of course, if you see a shadow of a hand, you don't know if it's a hand or just a clever rabbit. But anyway, in order to solve this ill-posed inverse problem, we have to make assumptions. Now, there's one assumption that is sometimes made and was very frequently made in the early days of MEG, when there were even fewer sensors in most arrays, was to assume a finite number of sources, usually one or two, and localize that one source very accurately. So for instance, and this works very well for some types of stimuli. So if you have an auditory stimulus, in this particular example, it was a click, and you can assume you have two sources, one in the left auditory cortex, one in the right auditory cortex, and you can actually localize that very accurately using one single dipolar or two dipolar sources. This can also work well for localization of the seizure focus if there are indeed only one or two discrete seizure foci. And in ideal cases, you can locate these sources to millimeter precision. But of course, the kinds of things that we're most interested in studying are generally far more complicated than something that can be distilled down to one single location in the brain. And, you know, given what we know about the brain, even for a simple source like a click, there are multiple processes going on as your brain is processing this, and it's a gross overstimulation to say that it's really just a single source. So instead of relying on individual dipoles, we can do something called distributed source model. And in our lab, we use a technique called beamforming, although there are multiple ways to do this inverse modeling. I'm not going to get into the mathematics here, but beamforming basically sets up a volumetric or surface-based grid And then for every point on that grid, the magnitude and the orientation of the source is calculated as a weighted sum of the data from each sensor channel. So in order to derive these weights mathematically, we need to know the field at the sensors generated by a unit source at each grid point. It's called the lead field, the covariance matrix of all the channels. And we calculate the weights for each grid point independently. Now, we also make the additional assumption that our solution is the one that minimizes power, minimizes the total power in the brain. So what can this beamforming technique do for us beyond, even for these, simple evoked responses beyond dipole localization? So to illustrate this, I'm going to show an example from a different evoked response study. So this figure shows the time-locked response to a phase stimulus measured with EEG. So there's a characteristic peak at 100 milliseconds that corresponds to the visual response. And then there's another peak at 170 milliseconds that's attributable to the fusiform phase response. In the literature, it's generally called the N170 because it's a negative potential and occurs around 170 milliseconds. But when we use MEG source localization, on the other hand, we can see that the spatial and temporal pattern of the phase response is actually much, much more complex than this. So this is a movie, working? Yeah. Done in collaboration with Carl Serrati's lab and two former postbacks. And this shows the response. And as you can see, the evoked response is actually quite complex. It moves from region to region. It evolves over time. And without adequate spatial resolution, the contribution from all these individual regions are averaged together, giving us what looks like a single peak at 170 milliseconds. But there actually isn't a single peak on 170 milliseconds. There's multiple peaks and multiple regions going on. And this is one of the things that we can use MEG and MEG source localization to show us. Nope, it's gotta stop that first, and then we'll advance. Okay. So when we talk about measuring brain signals, we usually talk about two different broad categories of responses that we look at in the brain. So I just showed you an example of an evoked response. So these are timed and phase locked to stimulus. But there's another type of evoked power response that I want to talk about. So the MEG power spectrum is broad. So this is just a power spectrum of magnetic brain activity or electromagnetic brain activity. And you can see the x-axis goes from 0 to 50 hertz. The spectrum actually extends way beyond that. There have been brain responses recorded up fast ripples, in particular in epilepsy, that go up to 200 or 300 hertz. But we divide the spectrum up into canonical bands. So delta, theta, alpha, beta, and gamma. And of course, it's a spectrum. There's not, clear delineations at the boundaries. However, there are some oscillations which represent very different dynamic processes that occur in assemblies of neurons. So while some activity, like those evoked responses, is time locked and phase locked to a stimulus, other responses represent an overall increase or decrease in oscillatory power in response to a stimulus. So this is just an example of a study looking using beamforming to look at that. So instead of looking at something time locked to a stimulus, we're looking. This was an in-back task, and we're looking at the oscillatory power following the stimulus presentation for a hard condition versus an easy condition in the in-back task. And this is in beta band limited power, and there's a decrease in beta power in the hard condition compared to the easy condition in central executive network areas that line up almost identically with what you would see in an fMRI study. We're actually using the same task in imaging and fMRI just to show that we can get comparable information for that. So this is an example of something that we can do in fMRI and MEG, that where we get a little bit more information when we do it in MEG, even though our spatial resolution is quite as good. So I'm showing the electrophysiology power spectrum again here, just to illustrate that point again, that we get a lot of new avenues opened up in functional imaging using MEG. So as a comparison up here, we have a power spectrum of a resting state fMRI time series. Now the different traces are from different denoising methods, which is irrelevant. But the important point is that the x-axis here runs from 0 to 0.18 hertz. So this is where the majority of frequency content in an fMRI time series comes from. So even though spatial resolution isn't as good in MEG, we still have vastly more temporal information encoded in the signal, and that can give us new insights. So this is another example using beamforming in band-limited portions of the spectrum. We can visualize the same resting state networks that are visible in resting state fMRI, but again, gain additional information on the underlying neuronal processes. So, and each of these blocks represents a different network, and on top is the fMRI, and on the bottom is the MEG. And the default mode network is best visualized in the alpha power band, while most of the other traditional resting state networks are best visualized in fluctuations in beta power. So this is an older study from 2009, but very pivotal, looking at resting state mag using an ICA method. But of course, now there are many different ways of extracting networks from MEG data. So again, to contrast MEG with EEG, we can look at functional connectomes. Of course, work in fMRI connectivity has largely moved away from ICA-based methods and more to functional connectomes. And this particular study was looking at connectomes in both MEG and EEG and comparing them. And you can see from these correlation matrices that, interregional connectivity is much higher in the EEG network. And this is largely the result of that smearing I was talking about. So you have these inflated correlations because the fields are getting conducted by the scalp. The MEG connectome, in contrast, shows much greater specificity and is likely to be more accurate into which regions are actually connected. Another point about using connectomes with MEG, not only can we look at connectivity in a single band, but we can look at connectivity across all the canonical bands, and we can also look between bands. So is there a correlation, is how power is fluctuating in one band correlated with how power is fluctuating in another band? So we did this study in collaboration with Carlos Zarate and a sample of healthy volunteers and participants with major depressive disorder. And there was indeed a shift in connectivity in MDD with decreases in alpha and beta connectivity and increases in delta and theta connectivity. So where the connectivity was occurring in the frequency spectrum seemed to be slowed in the depressed sample. And largely the same nodes that showed decreased alpha connectivity showed increased theta connectivity. We also looked at this same cohort after both ketamine and placebo, and ketamine robustly decreased connectivity, particularly beta band connectivity. So again, these two studies show that there's this extra layer of information that MEG can provide beyond just identifying what regions are functionally connected. So another commonly employed technique in MEG is decoding. And we can harness the high temporal resolution of MAG to determine at what times different low-level cognitive processes governing how our brain distinguishes stimuli occur. So this is an example from Bevel Conway's group at NIH and the National Eye Institute. And they looked at the accuracy of using MAG signals to decode color stimuli. And they used stimuli with different shades and with different luminance. And they found that, so this is a graph of the decoding accuracy over time. This is all colors averaged together, and here each trace corresponds to a different color. For whatever reason, pink was the best decoded. But there were important, and this occurred very fast, on a very fast time scale, between about 100 and 400 milliseconds post-stimulus. And there were also really important insights in this paper regarding how our brain views colors as more or less similar in terms of hue and luminance, and how those happen to differ for warm colors versus cool colors. So this is just an example of another way we can use imaging. To switch gears a little bit, I want to bring up a common myth about imaging, which is that MEG can't visualize deep sources. So it is absolutely true that the fields outside the head from deep sources in the brain are lower than those in the cortex. But that's not to say that they're not possible to measure. So on the left, we have simulated field distributions from the hippocampus, amygdala, and thalamus, as well as the neocortex. You can see that, and then on the right, we have the MEG sensitivity to those sources. And you can see that the sensitivity to the amygdala and hippocampus are only slightly less than that of the neocortex. And thus, this really, again, becomes a signal to noise problem. If you have an appropriately robust task and adequate signal averaging, these signals are measurable. And there have been numerous studies looking at MEG in the hippocampus and the amygdala. Thalamus is a little bit more difficult just because it's at the exact center of the brain. So several years ago, we wanted to demonstrate this, provided the task is appropriately robust. And this is work that was done by Linnea Forrest, who was postback in our lab, and Fred Carver, one of our staff scientists. So she adapted a reward task that incorporated low and medium reward and loss conditions, as well as an unexpected reward condition that we call the boost condition. So when we looked at this boost reward condition, we found that there were indeed decreases in beta power and increases in high gamma power in the left and right caudate. So we were absolutely able to demonstrate MEG signals from deep basal ganglia sources. Fred Carver also acquired data using a fine motor control task. It was a joystick center out reaching task, and found prominent high gamma activity in the target condition versus the control condition in the cerebellum. So taking these two studies together, we can demonstrate that it's absolutely possible to visualize deep sources using imaging. Okay. So to change gears a little bit, I want to talk about some of the things that are upcoming in that have become very important in the fMRI field. And one of these is big data, large-scale studies, and multi-site studies. Of course, there's studies like the Human Connectome Project, ADNI, the ABCD study, and all these have been done to encourage and enable reproducible neuroscience. And I want to talk about what we're doing in that space with MEG and what some of the hurdles and considerations are. First of all, MEG is a relatively rare resource. There are only about 40 or 50 labs or clinics in the US that have MEG, and there are really large swaths of the world that have no MEG centers at all, which is very different from MRI. There are MEG machines from 4 major manufacturers that are in use right now, and there's also a handful of other manufacturers who have only delivered a few machines. This isn't unique, of course. There's, Philips and GE and Siemens MRI platforms. However, the situation is a little different in MEG research. So the sensor geometry can be very different between the MEG manufacturers, and this is very important. So CTF, which is the system we have, uses axial gradiometers. So again, if you have a gradiometer, the signal you get is the difference between the field and the two loops. And so axial gradiometers are going to be most sensitive to the field that occurs immediately under the sensor. So in contrast, MEGAN, which is the primary, that has CTF and Megan together have the largest part of the imaging market share worldwide. And they use a combination of magnetometers and planar gradiometers. So these planar gradiometers have the current gradiometer loops on a plane, and they're completely insensitive to signals directly under the center of the sensor, and instead they're most sensitive to the signals on either side underneath the actual pickup coil. So if you look at topographical maps of the exact same brain activity from magnetometers and from planar gradiometers, it looks quite different. So this is an auditory task, and you can see a dipolar pattern in the magnetometers, but you see quite a different pattern in the planar gradiometers, again, because you're looking at the difference between those two sensors on the plane, and they're sampling different portions of the dipolar field.
Isn't the other way around? Isn't the other way around? The sensitivity of the sort of data.
No. So there's a dipolar field, right? You have a dipolar field from a single current source.
Yeah.
So when you look at the planar gradiometer, you're subtracting basically one part of the dipole, the positive dipolar field from the negative dipolar field. So you end up seeing something like this, instead of with the axial gradiometer, you're actually sampling the field under each sensor. You can, and generally what you do when you analyze this is the software will interpolate the fields for you, so you end up with this dipolar pattern, which is what's actually going on in the brain. But it's important to note that the underlying data is still fundamentally different. Sensor layouts are very different between systems, as well as the number of sensors. These are some MEG systems that are currently in use, and you can see that the layout, where they have gaps, how far down they go, is quite different. And remember when we do beamforming, we use a forward model from the MRI, and we derive these weighted sums of the sensors to solve for each point in the brain space. And a different distribution of sensors will give you a different sensitivity pattern across the cortex. It's going to be different for each vendor. On another note is that how noise is compensated is quite a bit different across the vendors. So our machine, a CTF machine, uses an array of reference sensors above the primary sensors, and it uses those to synthesize higher order gradiometers that are really good at eliminating noise, but other vendors use purely software filtering methods that have and there are advantages and disadvantages to both of these methods. So one thing we've been doing at NIH is forming a working group within the Enigma consortium. So Enigma was a project that was originally started to address reproducibility concerns and neuroimaging genetics research, but it's since extended well beyond that. And so now we have a working group that's devoted to demonstrating the feasibility of large-scale MEG studies. And we currently have about 41 sites worldwide that have signed on to this effort. And to show you an example of the kind of things that can be done, so on the left, this is one of the largest studies to date. It was just published in 2023 in PNAS, looking at lifespan and development effects on oscillatory activity in the brain. And so they found quadratic relationships between power and in different frequency bands with age. And of course, this was with about 300 or so subjects, and that was the largest sample to date that had been published in an MEG paper. Of course, we have a vastly larger sample. Well, I should say it's a larger sample produced in a study of this kind. I think there are other larger samples out there. But our sample is on the order of, it's over 1,000 right now and counting as we're adding more and more subjects in. And we can show more complex relationships. It's clear. This is the delta power in the middle temporal, which corresponds to, lost my mouse, there it is. Okay, so that basically is the same graph, the same data as this one right here. And while their data enabled them to make this quadratic fit, we could see, because we have a much richer sampling of the full time series, that it's really more of a cubic fit. And because we have such large numbers and we're adding more samples, we can start to look at things like eyes open versus eyes closed data, looking at genders, and in the future, looking at patient studies as well. So what is in store for the future of imaging? So, as I noted earlier, conventional imaging uses squid sensors, which need to be immersed in a bath of liquid helium to function. The thermal isolation means they're quite far away from the head, reducing signal to noise, not to mention the cost and difficulty with procuring liquid helium. We were supposed to have 250 liters delivered today for our cool down, and we got a invoice that said it had been back ordered. It's not easy to get liquid helium these days. It's coming tomorrow, so everything's fine, but it's a problem. So recently, optically pumped magnetometers have been developed, which operate on a totally different fundamental process, and they can be placed directly on the scalp. This has led to the development of numerous systems. So this image is from one of the very first really impactful OPM studies demonstrating the potential of wearable imaging. And since then, numerous whole head systems have been developed, although all of these have fewer sensors and sparser coverage than existing squid mag systems. It's still a nascent and emerging technology, really. So as I said, these sensors operate on a very different principle. The central portion of each OPM sensor is a cell containing a rubidium vapor. The vapor is polarized such that the opacity of the vapor to light depends upon the local magnetic field. There are coils that surround the cell. Losing my mouse. There are coils surrounding the cell on all the sides. I didn't show the ones front and back, but they're there. These coils null the field within the cell at the beginning of the measurement, and then during the measurement, they can be used in a feedback loop to keep the field in the cell along the sensing axis 0. Now, there are several challenges to using these sensors. The first is that the rubidium cells are actually heated. So we need to dissipate that heat somehow. Thermal management becomes a problem. There can be crosstalk between the coils on different sensors that has to be accounted for. And there's also an issue with ambient fields transverse to the sensing direction that we have to compensate for. But to tell you a little bit about what we're doing at NIH with these sensors, I'm going to return to this initial diagram that I showed earlier. So the technique with the highest spatial and temporal resolution is ECOG. Now that requires opening the skull and placing an electrode grid directly on the surface of the cortex, as shown over here. There's also stereo EEG, where depth electrodes are inserted into the brain. You can see this is a T where they show the depth electrode reaching down into the brain. And this is really only done in pre-surgical planning for epilepsy because it is such an invasive procedure. However, quite a lot of basic human neuroscience studies have utilized ECOG. So some of these studies come from studying unhealthy brains. So clearly a technique that has similar temporal and spatial resolution to ECOG, but is non-invasive, would be highly impactful, both for pre-surgical planning, but also for some of these human neuroscience, basic human neuroscience studies. So this is what we're planning to do, create a magnetocorticography device in contrast to an electrocorticography device with tightly packed sensors in an array that we can reposition anywhere on the head. So why is it important to sample densely and to sample close to the head for resolution? So the answer is in spatial frequencies. So any image, so any image is composed of a spectrum of spatial frequencies which encode the image. So low spatial frequencies right here encode coarse luminance variations, large object overall shapes, while the high spatial frequencies represent and encode the fine detail. So if we combine those spectrums of spatial frequencies, we get the high resolution image. So in the magnetic field, we look at a magnetic field pattern. If we have on scalp sensors and we can sample these higher spatial frequencies, we get a much finer resolution picture of the magnetic field, and that's much better localization. So this is some images of our device. Currently, it's not a whole brain array. The number of sensors required for a whole head array, this sensor density would be prohibitive at this point. Down here, this is a 3D rendering of some initial designs for the array. It's a little bit different now. But you can see there are heat sink fins here that are mounted on each sensor to dissipate the heat. The sensors slot into this 3D printed array. And there's a reference bridge here that holds 3 orthogonal OPM sensors that we can use for noise compensation. You can see here from the underside of it, how densely the sensors are packed. And this is our current setup, which will be changing very soon. But we have a subject wearing a neoprene cap that we attach the array to, and then the weight of the array and the associated cabling is supported by some bungees attached here to our SquidMag instrument. All of that will be changing. I'll show you that in a few minutes. So for our initial pilot work of this, we used a somatic sensory task where we had a stimulation device placed on the first, second, and third fingers, and we stimulated those fingers independently. We acquired data using both our OPM system as well as our CTF system. And these are just butterfly plots with the OPM measurements on top and the squid sensors on the bottom for four different subjects. And these are for all the stimuli combined. They're on the same scale, so you can see that there's a big boost in signal to noise that we get with our OPM array. And while the architecture of the overall response is the same between the squid and the OPM measurements, you can see there's a lot more detail in the response. Also, just because of the additional signal to noise that we get. These are some preliminary beamformer results for the primary response for each finger separately. I'll note that these are very preliminary. We haven't incorporated some of our calibration and co-registration refinement work, but you can see already that the result from the OPM array is much more focal than what we get with our SquidMag array. We also recently started piloting a tonotopic mapping task where we use auditory stimulation in one ear at 3 different frequencies, 300, 1200, and 4800 hertz. And we get a nice auditory evoked response here. And you can see the topo map here for each frequency. And when we use a classifier, we're able to discriminate between these 3 task conditions between 100 and 300 milliseconds post stimulus. So what MEG resources do we provide at the NIH? So apart from the research I've shown you, we keep the MEG system operational and all of our equipment upgraded. We do software maintenance and development. We do a huge portion of our time is devoted to user training. We also do data collection for shared data repositories and some MEG community support. Now, a note on training, importantly, because MEG is such a rare resource, frequently most of our users come to us with very little or even no experience in MEG, and we're happy to help fill in knowledge gaps if anyone is interested in doing MEG studies here. So we have a 275 channel CTF system in a shielded room. We have a full array, state-of-the-art stimulus delivery and stimulus response systems, as well as an eye tracking system. We use a brain site neuronavigation unit to co-register the MEG to the MRI. And we're also capable of doing simultaneous EEG acquisition. Our OPM system is moving. Fortunately, it will no longer be sharing a room with our primary MEG instrument. Instead, it will have its very own compact shielded room, which is being built right now and should be finished on Tuesday. Everything in the room will be finished on Tuesday, but the shielded room will be finished on Tuesday. This will greatly accelerate our research with this device, and it also means that we're going to be able to start opening up use of the OPM array to other users at the NIH, which we're very excited about. We're also currently part of the NIMH Research Volunteer Protocol. It's been in hiatus for a while, but it's going to reopen soon under Dr. Carlos Zarate. Under that protocol, we collected a comprehensive battery of tasks in MEG. There are currently 67 data sets available on OpenNeuro. The full set is 124, and that should be uploaded in the next few weeks. I think it's just about ready to go. They're just finalizing the upload. So if anyone is interested in getting their feet wet with an MEG data set, this is a great place to start. We already have some scripts and tools set up to help analyze this. We also run a ton of trainings and workshops. We've hosted trainings for both M&E Python and Human Neocortical Neurosolver. About every other year, we run the Mag North America meeting, which brings together researchers from across the US and Canada. We've also been running a Mag specific hackathon, and every year we've started doing an MEG boot camp in the fall. We will be doing that this year, although we haven't finalized the dates on that yet. And in addition to this, we have a lot of things like weekly journal clubs and other special lectures that we run from time to time. So thank you to everyone for attending. I hope it gave you some idea about what MEG is and what it has to offer, as well as a little overview of the resources available at the NIH. So thank you.